Hello everyone, myself SWAGATAM. This is actually the last part of my ongoing video series that is regarding AIDS. In my first video lecture, I have discussed that why the HIV is also called as retrovirus, and in my second video lecture, I have discussed that how exactly the HIV will get attached with its host cell by using different proteins that is present into the outer membrane of this virus. Now in today's lecture, basically I will going to discuss about that how exactly from a single HIV virus there form a huge number of HIV virus inside the human body to create the immunodeficiency syndrome. So let us have to start today's topic. So this is what the it's part 3, and today's discussion topic is formation of multiple numbers of HIV inside the human body. Now moving to the next slide. Let's suppose this is the whole HIV. This is the whole structures of HIV, and this one is the outer membrane of this HIV. Now if you guys have already seen my previous video lecture, that I have mentioned over there that the outer membrane of this HIV is constituted with special types of protein, that is the GP160, which I mentioned over here also, GP160. But the GP160 itself is a complex protein, and it is also constituted with two proteins, that is the GP41 and the GP120. The inner portion of this GP160 that is embedded into the outer membrane of this virus is known as the GP41, and the outer portions of this GP160 is known as the GP120. So this is whatever the structures of outer membrane and also the GP160. Now if I talk about the virus from inside to the outside, then before the outer membrane of this virus you will find another layer that is known as the matrix, and this matrix is actually constituted with the P17 proteins, which I mentioned over here, P17 proteins, and at the core of this virus you will find a special types of structure that is known as the capsid of this virus. Okay, this capsid is actually constituted the P7 and P24 protein, P7 and P24 protein, and into the capsid you will find the two single-stranded positive RNA along with the three important enzyme, that is the reverse transcriptase, integrase, and protease. So this is whatever the structures of HIV. The outer membrane that is constituted with GP160, and after that you will find a layer of matrix that is constituted the P17, and after that at the core of this virus you will find the capsid that is constituted the P7 and P24 along with the two single-stranded positive RNA, and also having the reverse transcriptase, integrase, and protease enzyme. Now once the HIV will get close with its host cell, host cell means some special types of cells where the virus will survive or increase its numbers, and the host cell of this virus is the CD4 plus cells that is present inside our human body. Once the virus will get closer with the host cell, the capsid which is present inside this virus will be diffused, and it will release into the cytosolic region of this CD4 plus cell. Now look at over here. Once the virus will get close with the CD4 plus cells, so this is the CD4 plus cells, and once the virus get close with the CD4 plus cell, it will get attached to the CD4 plus cells by using its the outer membrane protein, that is the GP160, and as soon as it will get attached, the whole capsid structure will be diffused into the cytosolic region of this CD4 plus cells. Once the capsid will enter into the cytosolic region of the CD4 plus cells, our cells, that is CD4 plus cells, will recognize this capsid as a foreign particles. Now to damage these foreign particles, the CD4 plus cells will release the protease enzymes, and by the protease activity the whole structure of capsid will be destroyed, will be damaged, and as the capsid will be destroyed, and now the remaining portion, that is the two single-stranded positive RNA along with a reverse transcriptase, integrase, and polymerase, protease, will release into the cytoplasm of this CD4 plus cells. Now I have to talk about the reverse transcriptase enzyme. There is the reverse transcriptase, and this is also the reverse transcriptase enzyme. Now this reverse transcriptase enzymes actually having the two activities. The first activity, that is the polymerase activity, and the second one, that is the RNase activity. Now by using the polymerase activity, By using the polymerase activity, the virus is actually able to synthesize the new proviral DNA. Now, question will be asked that was it necessary to synthesize the proviral DNA, because you know now that our genetic material is the double-stranded DNA. Now, to survive this virus, the virus will also have to convert its genetic material from the RNA to DNA. Otherwise, the virus will not able to survive inside the human cell, or it will not able to replicate its number in more and more. Okay. So once the virus will synthesize the proviral DNA by using the polymerase activity of this reverse transcriptase enzyme, immediately it will activate the RNase H activity of this reverse transcriptase enzyme. Now, by this RNase H activity of this reverse transcriptase enzyme, the old RNA strand will be get destroyed or will be damaged, and it will be released out from this cytosolic region of this CD4 plus cells. Now, the remaining proviral DNA will be diffused into the nuclear plasm area of the cytoplasm of this nucleus. This is the nuclear plasm, this is the nucleus, and inside the nucleus, the whole area is known as the nuclear plasm, because this is the cytoplasm of this nucleus. Okay. Now, I have to talk about the integrase enzyme. Integrase enzyme will also get diffused into the nuclear plasm. Now, this integrase enzyme also having the two activity. The first activity is a little bit similar with the endonuclease activity, and the second activity is a little bit similar with the ligase activity. Once the integrase enzyme will be bind with this with our human DNA, that is our human DNA, that is double-stranded DNA, and the integrase enzyme will bind with this double-stranded human DNA in its specific site, and by the endonuclease activity, it will make a cut into the human DNA into its specific sites. Okay. Now, this gap will be replaced by this proviral DNA. This gap will be replaced by this proviral DNA by the activity of the ligase activity, by the activity of this ligase of this integrase enzymes. As I mentioned that integrase having the two activity, the first one is the endonuclease, by which it actually made a cut into the double-stranded DNA, and now by using the ligase activity, the proviral DNA will get attached with this human DNA. Okay. Now, this proviral DNA actually having three important genes. This is the human DNA. This is also the human DNA, but the middle portion is the proviral DNA, and this proviral DNA, that is the proviral DNA. This proviral DNA also having the three important genes. The first gene is known as, this portion is known as the GAG gene, and the middle portion is known as, this one is known as the POL gene, and the remaining portions of this proviral DNA is known as the ENB gene. Now, once the proviral DNA will get attached to with the double-stranded human DNA, when the human DNA will enter into the central dogma process, will go through the central dogma process, the proviral DNA will also able to go through the central dogma process. Now, at the end of the central dogma process, that is after translation, the GAG gene of this proviral DNA will produce a structural protein, that is the capsid of this virus and the matrix protein, and the POL gene will produce the structural proteins, that is the reverse transcriptase, integrase, and protease, and the remaining portions of proviral DNA, that is the ENB gene, will be produce the GP160 protein. Remember, till now all of these proteins are structural protein, they are not functionally active. Now, after this translation, as soon as they will produce a huge numbers of GP160 and reverse transcriptase, integrase, protease, and a huge number of capsid and the matrix proteins, all of these structural protein will go through the post-translational modification, because post-translational modification is very much important to convert all of this structural protein into the functional protein. And ultimately, all of this structural protein will convert into the functional protein, and all of this functional protein is nothing but a huge numbers of HIV virus. So this is the sense through which from a single HIV virus, through the process of translation and post-translational modification by using the polymerase DNA that will form a massive numbers of HIV virus into the CD4 plus cells of human body to create the immunodeficiency syndrome. So I think this much